My name is Serge McGraw. I'm an associate professor at the Saint-Justine Research Center in Western Montreal in Canada. Many thanks to the organizers for giving me the opportunity to present our work today that was recently funded by the TBRS community. This first slide represents how DNA methylation in epigenetic modification is reprogrammed in early embryos. We see here that following fertilization, DNA methylation levels that are high will be lost. Then, once the embryo implants, the levels will be reacquired. So these levels of methylation are reacquired in a cell type specific manner during embryonic development and placental development. So in my lab, we study the early events that can lead to an abnormal programming of these epigenetic marks. We investigate how errors that could be produced in these early stages will be either generated and maintained or generated or corrected, and how the ones that are maintained can lead to developmental and neurodevelopmental disorders. We know from various studies that these early embryos, especially during preimplantation, if they do not acquire their proper methylation levels here at the implantation stage, these errors will be passed on in future cells. In the lab, we have 3 different projects or topics that are related to modeling errors in the embryonic epigenetic program. So the first one, we use embryonic stem cells to look into how specific genes can be altered and how they cause these epigenetic perturbation during development. In the second model, we have a preclinical fetal alcohol spectrum disorder mouse model to investigate how alcohol exposure will impact the early embryos during the first week of pregnancy, and how these errors that are caused on the epigenome of early embryos will lead to abnormal brain development. And important for this presentation, we are developing new models to solve how DNMT3 mutations affect development and cell specification. So in my lab, basically what we do is look at how the dysregulation of the embryonic epigenetic program can lead to developmental disorders. So I use the word epigenetic a lot, and you might have heard it before in scientific articles or in newspapers. Epigenetic is one of the hottest fields in life science at the moment, but what does it mean? Epigenetics is essentially additional information that can be layered on top of the DNA. So the DNA is composed of letters A, C, G, and T, and these epigenetic modifications will shape the actual appearance of the DNA. So these chemical modifications will act as switch that will order the DNA or genes to be turned off or turned on. So according to the modification, you can have a gene that will be on, and if it's another modification, the gene will be off. So when an embryo is fertilized, or when an oocyte is fertilized, this embryo is composed of early embryonic cells that once they start to specialize and differentiate, they can lead to more than 200 different cell types. And each of these cell types in these different tissues has an identical genome. The only thing that's different in these cell types is the epigenome. So it's the epigenome that will help to determine how the genome will be used. So for example, here we have an embryonic stem cell or a cell from an embryo, and once it starts to differentiate into, for example, here, a skin cell, during the differentiation process, the DNA from the cell type will acquire specific modification that will lead it to be a somatic skin cell. What is important to realize or to know is that the epigenome can be modulated and stimulated by outside events. Any outside stimulus that can be detected by the body has the potential to cause an epigenetic modification. It's not yet clear exactly which exposure will affect which epigenetic mark, and also what are the mechanism in the downstream effect. But what we're sure of is there's specific chemicals and lifestyle choices that can impact the epigenome. So across these different life events, we know that there's good environmental stimulus that can have a good influence on the epigenome, such as eating healthy food and doing exercises. This we know that it's good for the epigenome. However, we know that there's also negative stimulus, such as toxins, cigarette, different drugs that can have a negative impact on your epigenome. And we also know that epigenetic modifications, they are key during development and they need proper establishment. For example, here, across developmental time, there's specific genes that need to be switched on. So we need to have epigenetic modifications that keep this gene switched on across development. And this will lead to the normal development of the brain, the lungs, the heart, for example. But we also know that if these same genes have acquired abnormal epigenetic modification, this can also lead for the genes to be either switched off or have abnormal expression, and this will also lead to abnormal development of the different organs I just mentioned. A great example of how the impact of the epigenome can have an influence on the genome is by looking at differences that we sometimes observe in monozygotic twins. So monozygotic twins are babies that are developing with the same identical genome. However, at some point in their life, their environment might be different and the influence on the epigenome will be different. As such, you can have one child that at some point has some events in their life that can lead to cardiovascular disease, whereas the other baby has completely different set of life events and that baby will not have such a disorder. Similarly, we can have siblings, although they have different genomes, but they can have siblings with the same pathological mutation. However, their disabilities might be really divergent. And this is because, again, their life events might have been influencing their epigenome in a different way. There are many epigenetic modifications that can control gene expression. The main ones are histone modifications. So histones are small proteins, and these small proteins can be targeted by specific chemicals and will produce a more compact or more relaxed piece of DNA. So the histones are important in coiling or compacting the DNA to form chromosomes. And another modifications are the addition of methyl group directly onto the DNA. So my lab is particularly interested in these kinds of modification. They're called DNA methylation. So DNA methylation are added by DNA methyltransferase. They're targeting the cytosine in a CG context on the DNA, and they're really key in regulating gene expression. We're not going to talk about it today, but there's also DNA methylation involved in various processes that are implicated and absolutely necessary for proper development. So methylation is established by the DNA methyltransferase 3A and 3B. So these enzymes will add de novo methylation, so a new methylation mark on a naked strand of DNA. And then once the DNA is replicated, then these methyl marks that were installed by 3A and 3B need to be maintained, and this is the role of DNMT1. So in the case of DNMT3A, it acts together with another DNMT, DNMT3L. So we have complexes of DNMT3A and DNMT3L, so two proteins of two molecules of DNMT3A and two molecules of DNMT3L that form these complex that will produce these methylation tags on the DNA. And when we have mutations in DNMT3A, so depending where the mutation is in the different DNMT3A domains, well, what happens is you can impact either the formation of this complex, you can impact the way that DNMT3A will bind to the DNA, or because DNMT3A adds methylation through its methylation domain. So if you have mutation in that methylation domain, you might reduce, or you might even stop the DNMT3A from putting methylation on the DNA. So in kids with TBRS, we know that we have one allele that's mutated and one allele is fine. So what can happen? You can have complexes that are formed with normal DNMT3A, and you can have complexes that are formed with abnormal mutated DNMT3A and normal DNMT3A, and this will have an influence in the way that the methylation might be deposited on the DNA. So in my lab, our hypothesis is that the pathogenetic DNMT3A mutation lead to DNA methylation defects that will alter the normal programming of progenitor cells, so the early cells. And then this will have an impact on how the cells will differentiate and ultimately lead to the disease in the pathogenesis of TBRS. So about a year or two ago, we, with the help of clinical doctors and geneticists, we recruited a couple of TBRS patients, and one had a arginine 688 histidine mutation. And this particular patient really interested us because of the mutation was in the MTAs, and one important for DNA methylation, and once we use computational modeling, we also find out that the mutation was right at the intersection of the DNMT3A, DNMT3L binding, where they bind together, and also where they act on the DNA. So we wanted to model that mutation to really look into the mechanism that could lead to the pathogenesis. So we got a grant recently from the Rare Disease Model Mechanism Network and the TBRS community to model a specific patient-derived mutation, so the arginine 688 histidine, to define the timing and development that alter proper cell fate and development leading to TBRS-associated phenotypes. Because we, compared to genetics or genomics, where you could just take a swab in the mouth, or you can take blood and then study the genome, to actually study the epigenome, you actually need tissues at specific time points that you want to look at. So we decided to use a mouse model where we can investigate specific time points that are associated with specific developmental time points in the human, and use this mouse model to gather samples of the brain, samples of different organs, and look at how the mutation is affecting these specific cell types or tissues. So at the current moment, what we're doing, we're generating this patient-derived TBRS model. So we have, we're using the mouse, like I mentioned, and we're using CRISPR-based approach, like technology, where we can modify the genome of the mouse to introduce this specific mutation. And then what we're going to do is these mice are going to be breeded together, and then we're going to have mouse that have full mutation, so mutation of this specific site on both of their alleles, and then we'll also have mice that have a mutation on only one allele, and then we'll be able to define, how the level of the mutation, how it impacts development and brain development, for example. So across the development, again, we're going to be collecting different samples, and then we're going to be looking at the phenotype of these mice using different approaches. Also, we're going to be using imaging, and look at the epigenome and the transcriptome. So in terms of phenotype, we want to look at how the mutation will have an impact on the phenotype, meaning the morphology, the body weight, the bone length, as well as the brain size and different organs, and also look at the behavior. So we're going to be doing behavioral tests to look that are relevant to the neuropathologies observed in individuals with TBRS. For example, anxiety and memory. So different tests at different time points, either in young mice or in adult mice. And then we're going to be using a high resolution imaging to really look at sections of brains at different time points in development to really figure out what kind of cell type are affected. And this will be done to give us an indication on how brain function might be altered. So by using the behavioral analysis that we did before and combining these data with the imaging, we'll be able to define which specific neuron or interneuron, for example, is altered in the brain. And then what we're going to be doing is using these specific cell types, looking at DNA methylation and also gene expression errors that could lead to improper brain development and function. So we know that for brain development, we need specific set of methylation mark on specific set of genes in ways for these genes to be turned off. And we need genes that are lacking methylation marks because we need them to be expressed. So this will help us figure out how the regulation of these genes is altered and how this can lead to improper migration of brain cells and as well as lead to specific behavioral phenotypes. So globally, we expect that our model presenting a relevant DNMT3 mutation will show aspects associated with TBRS phenotypes that are not observed in current or cellular models because of the lack of competition between the wild type allele and the mutated protein. This approach will also help us understand the increased susceptibility of permanent impairments in the brain development and function associated with TBRS. And we hope in the future that our model, and not only this one, but other models that we can discuss later on, we can use these models to intervene on the activity of DNMT3A to improve the epigenetic regulation, the future cell fate and function, and potentially reduce some of the associated disabilities. So this is part of my small team. At the moment, we're about 10 people in the lab, and the two main people that work on this project is Karine Douaron, she's a postdoc student, and we have Alexandra Langford, which is a bioinformatician, and these are the two people that are driving these DNMT3A projects. And then we have a support team here of students and also team of researchers and geneticists that help us with patient recruitment, so specific aspects related to our project. So I hope this project has great interest for you guys, and I'll be happy to answer any of your questions or any of your comments. So thanks again for the invitation.